very, very pleased to invite onto stage today Erik Garrison, who's our keynote speaker for the event. It's an extremely difficult job to pick a keynote speaker for a conference like this, and lots of names were proposed, but I'm really delighted that, Erik, you're able to join us today. It's a real privilege to have you here, and I'm really excited to hear what you have to tell us. So I'm going to let you do your own intro pretty much.
Okay.
And thanks very much.
All right, thanks, Phil. And thank you all for having me here. It is a pleasure to get to give a keynote at to a conference that's representing such a diverse combination of people working in biology and biotech. And I guess the purpose of this talk is to look a little bit into the future about what happens with the kind of tooling and methods we're working with. And by tooling, I mean everything from the sequencing to the bioinformatic analyses. Yeah, I guess I'll give a little introduction of myself. I'm an assistant professor at the University of Tennessee, Health Science Center, which is in Memphis. And I've been working in biology for about 10 to 15 years. It's hard to say exactly how much was biology and how much was software tooling. But I basically came into it from following this belief that you should pursue the thing that's most weird and open in front of you. And that effectively led to this place. So first off, I want to give a little bit of technical background, because a lot of the things in the talk won't make sense if I don't provide a background to what pangenomes are and how we work with them. And then I'll talk about human pangenomics. So that's where we have developed a human pangenome that's designed to be an extension and growth and evolution of the reference system that many of you are probably familiar with. And then finally, there's a bit of biology that comes into this, because that system lets us see new things, and they're really interesting. So I'm really happy to get the opportunity to share them with you today. So the first, well, I guess I can give you a little more motivation to this. So when I first started working on actually looking at small variation against reference genomes, so I worked on a variant caller called freebase, I always thought that the most kind of fascinating future direction would be this problem of how do you understand variation between complete genomes? Like what happens when assembly is solved? Your default is you just know the genome of an organism, of an individual, of a cell, perhaps. How do you think about the variation between them? Because it's not just all point mutations, right? There's other stuff going on. And so the concept of a pangenome gets at that. I guess to the base level, a pangenome is just a collection of assemblies. So here, this model here, we have 4 genomes, and there's a maternal and a paternal haplotype for each. So they're diploids. And there's this kind of schematic representation saying, if you kind of align them against each other, you'd see that there's a deletion here, there's an insertion there, there's a point mutation somewhere else. And the idea is that those independent assemblies on their own, they aren't really enough to understand what's going on. So we have to put them into a model where that explains both their similarity and their differences. And a standard approach to do that now is this thing called a genome graph, variation graph, pangenome graph. I developed this concept of a variation graph. It actually combines the sequence graph with the genomes that walk through it. And so you can look at the graph and you see the annotations of the genomes through it and understand the variation between them. And so this is a cool thing because it's a compression of those other genomes. So maybe you could use it as a reference system on itself. You could align reads to it. You could think about variation in the population with it. And you can also visualize it. It's a human readable interface into variation between complete genomes. And so, yeah, this squiggle diagram at the bottom is kind of important because there's other ways of looking at this. This is one of them. So here you've got this deletion. So you could skip over a bit of sequence, the CAG, and we could represent that as well as a sort of string diagram where you've got a loop that you could go through, a bubble, or you could skip it. And that's exactly symmetric with an insertion. Indels they can go either way. They both look the same way, basically. But a point mutation, you would have an alternative. So you can go through a G, or you can go through A. And so you get a little bubble like you see on the right-hand side. Then this is a variation graph. This really represents the data model that we're working with. because you've got these paths that walk through those sequences, and if you follow the path and you read off the sequences on the nodes of this graph, then you get the genome that is being represented. You get the haplotype that's represented by it. So the idea with this model, the variation graph model, is that you've got alleles or sequences, and you have genomes going through it. And this puts all the genomes and all the sequences in them in relation to each other. So it's basically a solution to this fundamental problem in bioinformatics, which is how do you represent everything and understand how everything relates to everything else? And so it's this sort of ultimate bioinformatic object. It's very cool. And I spent about the last 10 years pursuing this topic, trying to first developing the model and then now applying it to data in the context of humans in particular. So the backstory here as well is that it's no longer a dream that you can have a complete genome assembly. Now it's just a matter of a bit of funds, because we have there's been this revolution in nanotechnology. We can see single DNA molecules, and we can see them with very high precision. And in fact, that's represented by this PacBio system, right? So in Pacific Biosciences, you can observe a single molecule, and you can observe it many times through a rolling circle amplification of the molecule. And those many observations let you get to very high accuracy. So you can have reads that are incredibly accurate. And that accuracy and the length of the reads is enough to sort of span most of the repeats in the genome, start to resolve everything, including the very complicated regions. But not quite everything, because you also have stuff that's so long and so repetitive that it's very hard to disentangle. And so the ability to see extremely long molecules through Oxford nanopore sequencing, that also, the combination of these two allows us to make complete assemblies, basically without trouble. And so it's becoming reality. Now we have this problem. We get lots of assemblies. How do we understand them? And this is also leading to the development of new resources, because we can make these assemblies. Now there's obvious push to extend the human reference so that it's not just one genome, but represents the diversity of the human population. And so I'll go over some results from this project. The Human Pangenome Reference Consortium, or HPRC, has been funded by the NIH in the United States to develop a reference system that is designed to replace the human reference and extend it. And we get a lot of authors, a lot of different interests, people working in production, people working in ethics, people working in sample collection, and people working downstream of assembling the genomes, and people working downstream of them. And we're mostly represented here. So we made a draft, right? So it's our first iteration. There'll be several iterations. The ultimate goal is to have something like 300 to 500 complete genome assemblies. This first one is just 47. And it has a very strange composition, because we needed to use something called trio-based assembly to make the assemblies. We wanted to be able to resolve every haplotype in every individual. So every maternal chromosome, every paternal chromosome, we wanted to separate them. And to do this, we needed trios. So we had to take cell lines, and we had to have cell lines, by the way, because sequencing technology works best on cell lines. And so we took 1,000 genome cell lines that had trios and had low cell line passage count and high diversity, and maybe had some unique alleles because they were more drifted from the center of human diversity. So that we ended up with this distribution that's mostly genomes from Africa and the Americas, with some from East Asia. It's kind of a strange mix, but I think in the future it'll be more balanced because of some of the requirements. We realize we can relax. So we get these 47 assemblies, and they approach the quality of the human reference. The Genome Reference Consortium's build, or H38, or I think it's build 19. It is like pretty remarkable what the sequencing technology allows us to do. So I'm skipping over the whole assembly part just to show that these are really good, and you can see that from this plot because it's showing you a bar that goes from 3 gigabase down to 0, and it's mostly green. And the green means that when we map reads from the assembly, when we map reads from the individual back to the assembly, it looks normal. It looks like it's properly haploid. We get both chromosomes. It's not collapsed. The coverage isn't weird. Only a small fraction, less than 1% of most assemblies, has any obvious issues based on this metric. And so they're really fantastic quality. And the contigs from these, they span whole chromosome arms, typically. So we're able to see really, really large portions of the genome that were invisible pretty much before this project. We'd seen, in some cases, one version in the T2T, that this is a complete human genome reference that was established recently. I'll mention that later. It's described over here on the right, but now we're seeing variation in those regions, right? So given these assemblies, we then wanted to make graphs to describe the variation in all the genomes. And there's 3 methods we used. The first is called minigraph. Minigraph is a progressive approach. It starts with one reference, and it layers things on top of that. And every layering adds a bit more sequence, makes a new graph, and then a new sequence can be aligned against that. So you basically take a series of assemblies, you order them in some way, and you do this sort of progressive alignment. And the result of it is actually that you get a model that's quite simple, which is very nice to work with, because it only encodes structural variation. So any new large sequences or large variation gets encoded in it without the difficulty of thinking about all the small variants, all those SNPs and indels that might each introduce new bubbles, new nodes, make your graph bigger and harder to work with. And so it has this very nice hierarchical kind of model as well that links everything back to the reference. And that can be useful for maintaining compatibility with existing tools. So the minigraph cactus model, this takes the minigraph and it adds the small variants back on top through an alignment. So you build the minigraph and then you align the reads back through that model, or rather, you align the whole genomes back through that model. And then cactus, which is a kind of multiple whole genome aligner. There's components in that are applied to then rederive the base level alignment. So we can see the SNPs and indels within the model. So this structure matches the minigraph, but then it has the SNPs and indels. It has a small variation inside of it. And so this, it has the same benefits and drawbacks as minigraph. It's progressive, so it's biased relative to the reference, but it does have the same nice hierarchy that links things back to a given reference genome. And then the model that I work on is kind of like the hard mode version. In effect, we align all the genomes to each other directly. And we do this with a technique, the tool is called WFMash, but we're linking a very fast homology search algorithm called MashMap with a very efficient way to do pairwise alignment called the wavefront algorithm. And those two things are brought together in a tool that can scale to really insane problems. Like this matrix that's shown over here, that's actually the alignment of all things homologous to chromosome 6 in primates, in great apes. And it's actually exascale. If you were to count all the bases in that matrix, it's quadrillions. And we can evaluate that very quickly. So we're able to do this comparison that's unbiased, right? So everything against everything else lets us see things without issue. And then this is converted into a graph in an induction step, this algorithm called Sequish. And then we refine the graph and smooth it. We make it simpler, make it more homogeneous with another tool called SmoothXG. And then there's a lot of downstream things that happen, including visualization, some of which I'll be showing you guys. Oh, I keep doing that. So we did these 3 methods because we couldn't really agree which one was best. But as a benefit of that was that we had cross-validation. So we had 2 ways to get to the same result, which was a variation graph that encodes all small variation, all large variation. And so we could extract invariant information. We could do cross-validation with reference-based methods to make sure that this variant calls were correct. And we could then look at the pictures of the pangenome that both of these methods drew. And we basically see that they're quite similar. I won't go too much into the details here, but they're both basically saying that we're adding hundreds of megabase pairs of polymorphic euchromatic sequence. So euchromatic being a kind of active regions of the genome where there's genes that are doing things that we think would be significant, not just repetitive sequences involved in centromeres or other satellite regions. Of those, there's a huge amount. It's almost an endless supply if you were to sequence more and more genomes. But the point is that you're getting 1 to 5 megabase pairs of new sequence per individual. It's a lot, and it's something we've been missing until we embarked on this study. So we use this to then look at interesting loci that are medically relevant. These are 2 that are quite important. One is the locus that defines the Rh blood group system. This is shown from the minigraph cactus model, and it basically, you can see there's these different haplotypes through the locus. There's quite a lot of diversity. That's shown in this figure C, the subpanel C. There's inversions and duplications of some of these genes. And I don't think that before we had these kind of complete assemblies, there hadn't really been this deep survey of this region. And now we're doing it everywhere. Here's another example from the human MHC. This is around HLA-A, and it has a lot of the pseudogenes that are related to it. This is from the PGGB graph, and we showed a bit of both. One interesting thing is that this region around HLA-Y, that's an ancestral sequence. It's quite large. I think it's in the hundreds of kb. And it's not in the reference, actually. It's deleted from the reference. So there are a few sequences like this that were just being left out entirely. They're unique, and we didn't see them before. So this is kind of showing you that you can zoom in from all levels and understand variation at all scales. So at the bottom, you've got one of these kind of string diagrams of the graph. This is the P arm of chromosome 6. And there's a centromere over on the right-hand side, which is this tangled loop, which is quite repetitive. It's kind of sort of a variable number of tandem repeats. It makes a loop, and when you build this sort of graph. And then the MHC is this locus. You can actually see it at the scale of the centromere. There's this big bubble. And when you zoom in, that's actually the MHC class 2 gene, HLA-D and friends, D star, I should say, all the different HLA-D versions and the different haplotypes associated with them, they make this big bubble. And one locus we looked at quite a lot was one called C4A, C4A and B. And that actually forms this kind of loop as well. So there's this particular graph building technique will tend to collapse polymorphic repeats into single loops. So you have one base version of that repetitive sequence, and every genome goes through it multiple times. So this locus is interesting biologically because it's been implicated in schizophrenia risk. There's some contest about that implication, but it's certainly important in the immune system, and it is variable in the human population. People have 1 to maybe 4 or 5 copies typically per haplotype. And one thing that's really nice about this locus is it exposes something that we see because we can make these complete assemblies, which is that there are very unexpected, nonlinear kinds of effects in genome evolution. And what I mean by that, I have to describe a little bit what this figure on the right is to explain. In effect, if you were to take the reference C4A and C4B, and then you take every haplotype through this loop, and you ask, can you deconvolve me relative to the reference? Like, tell me which of those genes I'm more similar to in what order. You get this sort of plot over here on the right. And you can see that some haplotypes have like 1 copy and some have 3. Most have 2. But there's something really fascinating that comes out of a few haplotypes, which is that instead of going C4A and C4B, they go C4B and A. And so that means somehow information was getting mixed up in a kind of nonlinear way. And we think, and actually there's a companion paper at the HPRC, that this is being caused by gene conversion. So this is actually a kind of a rampant process in the genome that, homogenization is perhaps the most important kind of recombination going on, but it's been very hard to see if you look at populations with short reads. Okay, so that's a whirlwind tour of a draft human pangenome. So now I want to talk about something new we see, something a part of the genome we couldn't look at before, where we can now understand what's going on. And the subtext is we finished the human genome, right? We actually got a T-to-T, telomere-to-telomere assembly of a single human genome. It was made from what's called a hydidiform mole, which is kind of a pseudo-haploid human cell line that had been extracted from this. It's called CHM13. And this actually filled 8% of the human reference that was incomplete. It's mostly centromeric. You can see in this karyotype, the black bars, those are regions we didn't have in the reference. But also the acrocentric short arms, and that's what we'll be focusing on here. Acrocentric chromosome is a chromosome where the centromere is really close to one end. So it has a very short arm and a very long arm. And in humans, all the acrocentrics, they're the locus that codes for ribosomal DNA, which is, in fact, the ribosome biogenesis is the most energetically intensive activity of the cell. So it's a very, very important, and the fact that they all encode for it means that there's this kind of duplication and redundancy, but it also means that these sequences were very hard to resolve because they must be somewhat similar. And so there kind of is a new mystery that comes out of this. And this paper gets into it. I don't have much time to show details of it because I'll have other ways of showing it to you. But when you make the assembly graph out of the hi-fi data for this sample, every single chromosome makes its own component except for the acrocentrics. All 5 of them are involved in one graph component that's isolatable. And that was very suggestive. I think people thought that it was the ribosomal DNA, which is repetitive, that was causing this to happen. But then we also saw it in the HPRC. Our assemblies don't really assemble the ribosomal DNA because it's very, very long repeats and very hard to assemble through. So something else must have been going on. Basically what we're seeing here are, you take the T to C assembly, right, and align each of our HPRC haplotypes against it, and ask when the haplotype switches affinity between reference chromosomes. So switching between reference chromosomes might usually say that seems very unlikely biologically. Like it's weird if you have it, it must be like a cell line artifact, right? You've got two chromosomes fused together or mixed in some way. And we saw maybe 1 or 2 cases like this, but the vast majority of the actual genomes showed this signature of having contigs from the assembly that mixed different acrocentric short arms. And actually contigs, I should note that there's a bit of a jargon from the assembly world. Contig refers to a contiguous set of sequencing reads, like a part of the assembly that you get from overlapping the reads that you're really confident in. And in the HPRC, that's our main currency, because we said, well, we'll take the contigs because we don't really know how they fit together at a higher level. We can see from the perfect overlap of the reads that these are something we can trust. So that's a contig. And we're going to take these contigs. So based on this observation that there's this weird mishmash of things happening, we thought, well, we'll take all the contigs and we'll run the same kind of analysis you see here on the left, where every contig is a node and the edges represent mappings between them. And so here the reference contigs are circled in color, and you can see everything else kind of mixing between them. So we'll do that for all of the contigs and see if, is this happening elsewhere? Like what's basically going on? And so we do this and we get the same kind of figure. This is made by Gephi. It's not like scientifically accurate. It's for human visualization. But the distances are suggestive. And so you see the sex chromosomes form a little cluster, and the acrocentric ones make the same kind of cluster up there that, you have the contigs from the Q arm, the long arm of all these chromosomes. They're sort of on the edge. And then in the middle, if you look, it's stuff that maps to the short arm. And it's this big mix of different colors. And the colors we've assigned based on like our best guess of where the contig comes from, but they're clearly unreliable, right? Like they're mixed up. And so then we thought to do a quantitative approach to understand what's going on. We applied a community detection algorithm. We heard a bit about this in the talk about molecular pixelization of cells. So we use this modularity maximizing partition of the same graph I was showing you to break it into parts that are kind of self-contained. And every chromosome or chromosome arm is self-contained except for the sex chromosomes, which you can see kind of at the bottom right. There's the chromosomes at the bottom and the community is up here. So there's multiple things in one community from different chromosomes. And of course, the acrocentrics. And if you look at the actual partitioning, you can see what's going on, that there's this kind of single community representing a lot of the acrocentric cluster. That was very suggestive, but we can make a graph that shows us the exact base pair relationship of these genomes. And so we took the best assemblies from these regions, from the HPRC, and we found ones that found contigs that overlap the centromere. So they go from a part of the chromosome we think is unique, the long arm, into a part that we think is getting mixed up, the short arm. So we take all of these, and of course, what do? We make a graph using PGGP, and that results in this image. Well, we render it too, right? We make a visualization to get this image. And so the graph has been laid out in such a way that the distances in these 2 dimensions, they match the distances in the contigs that are inside of it and the haplotypes that are there. And we colored things based on where the T to T chromosomes fit inside of it. And you see that the Q arms are separated if we were to zoom in on this box, you see in B, there's this very specific relationship between the chromosomes that 15 is a kind of an outlier, but 21 and 13, they effectively share a centromere, and then everything after it. And the same is true for 14 and 22. And then they come together in this complicated mess. It's actually very hard to disentangle. So we decided to try to disentangle it to understand what's going on, right? Because this is not enough. This is like a kind of population cytogenetic view of what's going on, but it's still just 2 dimensions. It's hard to see. So, is this caused by some kind of homology that's ancestral, or is it something maintained by ongoing recombination? It'd be very exciting. So we untangle the pangenome graph, and that basically involves, for every contig, which are shown here as rows on this plot, at every part of the contig, we're going to ask, what reference chromosome is it most similar to? And so here we've zoomed in chromosome 13 contigs. They're on the short arm, and you can see the long arm is over here. Everyone matches 13. And then there's the centromere. No one matches anyone, because centromeres are all diversifying, and it's very hard to actually derive this kind of untangling there. And then there's this region, which it's labeled SST1. That's a repetitive sequence in the middle of it. But every single contig is like a patchwork in this region. They're all mixes. And they're specifically mixes of chromosome 13, 14, and 21. And then if you get closer to the ribosomal DNA, there's some other stuff going on with 15. So it starts to play a role. And so to show where this fits on that 2-dimensional plot, that would be this kind of long arm, right? The centromere is this sort of hairball loop where no one is like anyone else. And then this region around SST1 kind of it's really hard to see on the plot, actually, but it's here. And then there's this ribosomal repeat, which we can't assemble through. Only the T-to-T assembly has any representation of it. And so these regions, actually, I skipped a bit. So we build a kind of entropy metric. We have a quantitative way of doing that that's based on this sort of patchwork pattern you see. Excuse me. And we use that to define what we're called pseudo-homologous regions. So these are places where multiple chromosomes are mixing, and they look like they're, hanging out together. And it gives us this sort of intervals across the reference. We get a new annotation type. And we can do this for all the 5 chromosomes. So for 14, you have a similar kind of pattern. Actually, stuff's going on even before the centromere, in this case. It likes to match up with 22. 15, like I said, is a bit of an outlier, but close to the ribosomal DNA, it starts to behave the same. 21, it's almost a mirror of what you see in chromosome 13. And 22, again, it's a little bit different than the 3 kind of main ones involved in this process, but it also has its pseudo-homologous regions where there's exchanges. So, then we wanted to zoom in on this particular region. It's very interesting, between 13, 14, and 21. The interesting thing about this, the suggestive thing, is that chromosomes 13, 14, and 21 are involved in what are known as Robertsonian translocations. These happen about 1 in every 800 births, and they happen de novo. So there's basically recombination between 2 chromosomes, and the Robertsonian chromosome is one that mixes 2 of these acrocentrics. There's 2 centromeres, and it's lost its ribosomal DNA. And this region looked to be a potential place where that might be happening. And one of the suggestive things is that the region is inverted on chromosome 14 in most individuals relative to 21 and 13. So there's a lot going on here. I'm going to just mention that there's a repeat in the middle called SST1, and we looked at the repeat units of it. This repetitive sequence exists on other chromosomes. But if you make a phylogeny of them, every chromosome is like monophyletic, basically. It's its own clade, except for 13, 14, and 21. And so there must be some exchange driving that. And then we dug in with a different way. We looked at older data. There was a fish mapping experiment on Robertsonian chromosomes from 14 and 21. They took probes from the GRC. They took the backs, actually, that were used to make the assembly. And they saw where they fit on the Robertsonian. So if they're present, they're green. If they're not present, they're red. These 2 plots. And so what we're actually showing is a hypothesis for how the chromosomes match together to make the Robertsonian occur. And given this hypothesis, it's sort of completely consistent with the fish mapping data. So the green bits that you see And by the way, there's none on 21 because they used the chromosome paint to actually label the Q arm there. You know, it would match recombination in this region. And the fact that you don't find anything on the short arms, this red that you see on the sides, is consistent. And so that there's a model of this that's shown below. Basically, if you have this recombination, where 14 is inverted relative to 21, you'll get this microchromosome. It has no centromere, and it'll be lost. And it has the ribosomal DNA on it twice, but it doesn't continue. And then what actually is passed on in the cell line in the individual is this fused chromosome. So this is pretty fun because Robertsonians have been known around for a long time because you can see them using cytogenetic techniques that have been available for 50, 60 years. And so the question about what's going on here was very hard to understand, though, because you can see the result, but you can't see the process, and you can't see the details of ongoing processes you might be maintaining it. So it was brought up in this paper that actually was the first to sequence the centromeric repeats on these chromosomes. They got the sequence of the satellite repeat, and they said, wow, these chromosome 13, 14, 21, they're so similar that probably there's some ongoing recombination exchange between them to maintain that similarity. But that's kind of where the story stops. Everyone says, okay, makes sense. This is another example from the 1970s. Very laborious process. A spermatocyte has been frozen, fixed at what's called packetine. That's when the maternal and paternal chromosomes have been pulled together and the recombination is occurring. And slices are made through this, and they do an electron micrograph of them, and they're layered, actually layered them with like sheets of transparency to make a sort of pseudo 3D image, which they then condense into this drawing, in fact. And what's really fun about this is that the nucleoli are these black things. And there's one nucleolus, it has 3 chromosomes on it. And you can guess which they are. They're 13, 14, and 21. So this is an N of 1 thing, but it would look like, this is N of 1, they only did like 2 or 3 of these. So the sample size wasn't very big, but there was a lot of cases where this was occurring in effect. And so they're hanging out together, right? The chromosomes are close physically, which is a precondition for recombination. And this is to show that a direct ascertainment of this is extremely difficult. So if you want to say, find a case where this recombination is occurring, like in the act, what do? You get oocytes, where you think that the Robertsonians occur, hundreds of them. They use 300. And then you do FISH to try to color the chromosomes with fluorescent signal. They find one case for 14 and 21, look like they're at packetine and mixing. So 1 in 300. And that's actually very close to the rate of Robertsonian translocation. So the problem is that to establish, strongly that this is going on, you might have to do 10, 20 times that, and it's just not feasible. So we had the benefit of an amazing resource, Human Draft Pangenome, that lets us look back in time at the human population and do this kind of population genetic analysis of variation of whole chromosomes. And that gives us this insight that there's a region where there must be recombination happening. So there are these pseudo-homologous regions on the short arms of the acrocentrics. And we think that these occur due to physical proximity. Right? The nucleolus is kind of containing the ends of the chromosomes that are associated with the ribosomes. And so they're close together, which gives them the opportunity to recombine. And they have a similar function, too, so they should be similar. So most recombination is actually conversion. About 90% of double-stranded breaks will resolve as a gene conversion event. So this homogenizes the chromosomes relative to each other. We know, based on the fact that Robertsonians exist, that some of these must be crossover, and because how else could you get a Robertsonian chromosome? And so that leads to the synthesis of what's going on and perspective of a region of the genome that was invisible until a few years ago. So we published this alongside the main HPRC paper in Nature in May this year. And it's been an amazing journey to be on, in fact. And I hope it's interesting to you guys to think about what we might see in the future. So I want to thank a lot of the folks who've been involved in this project, in particular Andrea Guaracino, who did a lot of the hard work required to bring the project to conclusion, and also the whole Human Pangenome Reference Consortium, which made this possible fundamentally. So with that, I'd be happy to answer any questions.
Thank you very much. It was a fascinating story. It was really good. And ties in very neatly. I should little plug for the Boston Summit we have next month where Adam will be talking.
That's
He's our keynote speaker.
Adam is an important collaborator on this work, and we're actually I didn't mention this, but we're assembling Robertsonian cell lines, and we do have confirmation that that is the region where they recombine. So with Adam,
yeah, we've got lots of questions here. I'm going to pick a couple. Firstly, one from Geraldine. Do you envisage running analyses like variant calling directly on pangenomes as references? And if so, do you think we can still output VCFs, or do we need new formats to represent results?
Yeah, so we can output VCFs. So actually all the work we did in HPRC is sort of ironic. A lot of it feeds back into VCFs against the reference. Even the alignment and variant calling workflows that we presented, they always made a VCF against the reference. So we didn't really explore the full benefit of working on the graph because we lacked the infrastructure, the kind of design, the community infrastructure, of people understanding how to describe variation against the graph. We have models for it, we have data formats, and I think it's completely possible. We have draft versions of ways to do it. But that's interesting. I do want to point out that there also are new opportunities to simplify thinking about variation. So the fact that you have all the alleles that you might be interested in means that you can just jump right into a matrix of allele counts. And that's something very similar to what we work with already in population genetics and GWAS. And I think that could be a way to simplify, kind of cut through a Gordian knot of complex pipelines that we've developed over the past years.
Kind of related to that, maybe unsurprising given the audience, there's an interest in tooling and
Yes.
in building on this.
Yeah.
Abhinav asks, to what extent are current tools compatible for a variation graph-based reference?
Yeah, that's an interesting question. So, in one perspective, nothing works anymore. You say, oh, now I have a graph as my reference, and all is lost. I should rebuild from nothing. And I think that's not accurate because we put all these genomes into that graph. So we actually have many, many frames of reference to work with. And that's part of the basis of our work, was to use those different frames of reference, not just the graph. So any annotations you have against a genome, they can be put into the graph space. They can be injected into that context. And any tools that work on the genomes themselves, they have a natural basis in the graph in that way. So the pangenome is not just graphs, it's really the genomes, and maybe the bigger problem is thinking about just how to work with lots and lots of genomes. Then all the existing tooling we have will continue to work. And we have to do that, and then find ways to harmonize it with the graphs, not necessarily rebuild everything in the graph space.
Last one on tooling. Samuel asks, have you been looking into interoperability between variation graphs and tree sequence data structure used by toolkits like TS-Kit?
Tree sequence data structure, so...
It says it can link different genealogical and phylogenetic trees to different parts of a genome due to the different histories across the sequence.
Right, and this is related to maybe ancestral recombination graphs. I've wanted to find a way to link the two. I guess it's possible in the sense, based on my previous answer, you've got genomes and you've got ways of working across those coordinate systems. So in principle, we can do that. We can get variants out of the graph in reference coordinates that can be usable by these existing systems. A deeper integration, though, that would really take someone who loves the two topics.
Ricardo asks, how do you represent rearrangements across different chromosomes, and are they independent haplotypes between them? Do you have any ideas on how to do this with polyploids with complex rearrangements?
Okay, well, so we put a bunch of genomes together, right? So polyploids, no problem, right? You can have one haplotype, you can have 10, it's going to work out. You just have to remember which ones they are. And so remembering which ones they are may be as simple as like naming your sequences appropriately. So there's that part. I think that's okay. What does a rearrangement look like? it's going to be a case, like in the graph, it'll merge different chromosomes together. Because there'll be a sequence that goes from what's typically one chromosome into another. And so you can see that by maybe untangling, or actually by looking at the structure of these two-dimensional layouts or embeddings that can be produced from the graphs. So they sort of naturally work. The only problem is that as you get more and more rearrangements, for example, in a case like in prokaryotes, in bacteria, like E. coli, that have very open pangenomes, then everything is just a hairball. And when you try to look at it in low dimensions, you're completely confused. So yeah, the model represents it, but it may be hard for human eyes to see. Yeah.
Last question from the audience. Hashor says, as a guesstimate, how many human genomes would we need to do to sample to get a good gauge of genome diversity in the human population?
Okay, well, yeah, it's a standard question, right? Like, I guess you can always go down in frequency because we have this rapidly growing population. There's a lot of rare alleles, and so you'll always find something new. And it depends a little bit what part of the genome you want to look at. So what we learned in the HPRC was that the euchromatic sequence is homogenizing, right? And it's under some constraint. So you might have to see fewer genomes to see that. And you would kind of saturate eventually. You'd find most variants. You'd see maybe not every SNP, but you'd find most large variants or recurrent variants. And this is not true for the satellite sequences and the centromeric sequences. Those are under diversifying selection and they're like these arms races with each other to be better at being centromeres. And so the more genomes you see, the more centromeres you see. You'll see very big sequences that are, they're not unique in maybe components, like they're made from the same repeat sequences, but the arrangement will be completely unique. And so you could continue doing that forever through the whole human population. But I think that the standard kind of ideas from biomedical informatics apply. So you do 1,000 genomes, you're going to resolve almost anything that you're going to see in anyone. Okay, let's leave the centromeres aside. And you do 100,000, then you really start to cover everything. Maybe almost every base has a mutation, and you learn a lot from that. So I guess this is really an endless process. We've begun. There could be a new pangenome for every population, for the whole human population, for every generation even. Our composition as a species will change over time. So yeah, it's the beginning of something similar to what we're doing, but very expansive in scope.
Cool. My final thought is I think it's a really interesting discussion happening on Slack. There's lots of kind of impressions. And my first taste of this was when Simon came and asked about putting a representation of this into multi-QC. And he showed me these beautiful images of string diagrams. And I was like, and I think everyone is kind of sitting there thinking, is this the future of genomics? And is this going to be a watershed moment where we look back on how we used to do bioinformatics and genomics and takes us forward to a more accurate, better representation? I guess, you're biased, but you guess this is the future.
Okay, it's like, yeah, it's a certain kind of watershed. I think that confusingly, all things will continue. So we'll continue to get short reads because they're super cheap. And we can then use them against this system to do better. But the short reads will exist, right? We still have array data because it's really cheap. Because all the things that you got good at doing in the past, they become very low cost and they have their utility. So I feel like this is just an expansion of the kind of scope that genomics can have, rather than a replacement. And it's not like things that we've been doing in the past become irrelevant.
Thank you very much. Thank you for giving us a glimpse to the future.
Yeah. Thank you. Thank you so much. Thank you all.